Thank you.
Hi everyone, so we'll get started in just a few minutes. For now, please make sure everyone fills out the attendance form that's in the chat, and we'll get started shortly. Thank you for waiting. Hi everyone, we'll get started in just a minute. If you haven't already, please fill out the attendance form so we know that you were here. And yeah, we'll get started in just a moment. Thank you so much for waiting.
Hello everyone. My name is Sharnam Chowdhury. Be your tutor for today, so I'll be going over the study session for exam 2. Obviously, we are limited in time. I'm sure if some of you guys have started studying, you know it's a lot of material to cover just during free hour. So I'll try my best to get through as much material as I can. And if it isn't possible to get through all the material, don't worry, because the slides I have, the slides are going to be posted on the slides are going to be posted on the Queens College Women's website, so you guys would be able to see the slides later on. So if we're not able to go through all the slides, don't worry. You'll be able to see the slides on the Learning Commons website. So I'll start right away because like it is a lot of material. It's very dense. Um, all right. So are you guys able to see my slide? Just say yes in chat or put a thumbs up or
Yep.
something. Yeah. All right. One thing also I'll say is that we're going to be going over the main concepts. Because we're going over the main concepts, obviously you guys have to do your own supplemental studying. Stuff that I don't go over in these slides, you're still gonna have to do. So for that, definitely look at Dr. Fath and Dr. Garofalo's slides because they'll be your best resource. All right, so I'll start off with cellular respiration. All right, so cellular respiration, we start off with obviously. So the sun, for plants, they go through photosynthesis, and then through photosynthesis, they produce glucose. For humans, obviously, food, and then through that food, we get glucose. That glucose eventually goes through glycolysis. The glycolysis creates pyruvate, and then from pyruvate, we can either get aerobic cellular respiration or anaerobic respiration and fermentation. So glycolysis, for like the Latin, it means sweet, and then unfastening. So for that, if you know some Latin, obviously it'll help for some of the words. All right, so glycolysis is the breakdown or oxidation of glucose to pyruvic acid. So oxidation means the loss of electrons, so something is becoming more positive. That's another thing I expect you guys to know, redox reactions over time. Um, so like I said, glycolysis is the oxidation of glucose to pyruvic acid. So for step one, ATP loses a phosphate, turns into ADP. So it uses up ATP. ATP gets hydrolyzed, and two of the first three steps require energy for the hydrolysis of ATP. So you have your molecule of glucose, that requires energy from ATP hydrolysis, and that eventually produces glucose 1,6 biphosphate or bisphosphate. Then eventually that glucose 1,6 bisphosphate turns into what we could know as G3P. And so G3P is important because G3P also creates glycerol, which is normally seen in your fatty acids. So after that, the G3P, um, after the G3P gets used, now ADP turns into ATP. So now you're producing ATP. ADP combines with another phosphate to produce ATP. So we get two ATPs here and four ATPs here. And then as NAD plus takes in a hydrogen, eventually that G3P turns into pyruvate. So the main steps for this that you have to know for glycolysis, glucose uses up ATP. So ATP gets hydrolyzed, the glucose uses up that ATP to turns into fructose 1,6 bisphosphate. Then eventually that fructose 1,6 bisphosphate turns into G3P, and then G3P turns into pyruvate, where NADP plus gets used up to create NADH, and ADP combines with the phosphate to produce ATP. to finally get pyruvate. So that's as you can see here. So we use up 2 molecules of ATP during glycolysis, but as you see here, we gain 4 molecules of ATP as you see through the red circles. Use up 2 ATP. We gain 4 ATP. So our net gain from glycolysis is 2 ATP, and we also produce pyruvate molecules. So now I'm going to talk about fermentation. So fermentation is essentially trying to replenish the pyruvate or replenish the NADP plus so you can keep the process of ATP going. So as I mentioned before, fermentation only produces 2 ATP. Cellular respiration, which is the main way we get energy, produces 32 ATP. So for certain bacteria, they use fermentation. So some cells use fermentation to regenerate NADP plus to keep glycolysis going. Are you guys able to see my mouse?
Yes.
Okay. So some cells use fermentation to regenerate NADP plus. So here, right here is where the step of where glycolysis ends. Pyruvate gets formed. So for fermentation, pyruvate eventually turns into acetaldehyde, and acetaldehyde then oxidizes NADH to produce NAD plus. And then eventually that acetaldehyde turns into ethanol. So that NADP plus, as you see here, it's a cycle. It goes right back to where we started with glucose. NADP plus turns into NADH, and that glucose eventually turns into pyruvate, producing a net gain of 2 ATP. So for fermentation, it is a cycle where glycolysis essentially happens over and over again. So that way you keep producing a net gain of 2 ATP. So now we're going to talk about pyruvate oxidation as you see in cellular respiration. So pyruvate eventually turns into acetyl-CoA. So a decarboxylation of each molecule of pyruvate yields 1 molecule of carbon dioxide and acetate. And then that acetate eventually is used to produce acetyl-CoA through coenzyme A. So pyruvate turns into acetyl-CoA. So that's pretty much the majority of what you have to know for pyruvate oxidation. Just you don't need to know the exact specifics, just that pyruvate turns into acetyl-CoA through coenzyme A. And NADP plus gets used to produce NADH, and carbon dioxide gets produced as a byproduct during this time. So now we're going to talk about the Krebs cycle, or also citric acid cycle. So as you see here, it is a lot of information, but you only need to know the main steps. The main steps is that acetyl-CoA is input, and then acetyl-CoA gets oxidized. After acetyl-CoA gets oxidized, NADH gets formed, through NADP plus, carbon dioxide gets released, and then eventually that acetyl-CoA releases carbon dioxide, produces a small amount of GTP, but its main function is to get NADH and FADH2, which are electron carriers. So that's the main function of the citric acid cycle. You input acetyl-CoA, water, and G deep, and eventually, later on, you're able to form NADH, and then later on, you're also able to form FADH2. So these electron carriers are important because eventually we'll go through the electron transport chain because that's how you're able to produce ATP. So main function of citric acid cycle, once again, you produce NADH and FADH2, which are the reduced electron carriers, so that they could send the electron to the electron transport chain in order to produce. So now we're going to go over the electron transport chain where you produce ATP. So an electron is transferred from NADH and FADH2, which you obtained from the citric acid cycle. And then you have redox reactions where each station oxidizes the previous station, and then that station becomes reduced. So electrons are accepted by NADHQ reductase at the start of the electron transfer chain, and electrons obviously also come from FADH2. So as you see here, these electron carriers send an electron down the um to the stations, each station of that you see here, and then eventually these electrons keep getting sent down to more complexes, cytochrome C reductase complex, cytochrome C oxidase complex, and eventually the final electron acceptor is oxygen. So as you see here, NADH gives an electron to NADHQ reductase. FADH2 gives an electron to ubiquinone. And then the main thing here important is that as they give an electron, it creates a proton gradient at the top. So this proton gradient is important because eventually the protons are going to go down ATP synthase, which obviously spins the ATP, producing ATP at the end. So once again, NADH and FADH2, which you received from the citric acid cycle, they send an electron over to these complexes. Hydrogen goes through, or H plus ions go through into the intermembrane space. It creates a proton gradient where you have an extremely high H plus concentration and a positive charge. So you need to get rid of that positive charge. You can't have too much of a positive charge in your intermembrane space for a long period of time. So because of that, hydrogen ions then go through the ATP synthase in order to reduce the proton gradient that you see. Hydrogen ions go through the ATP synthase. The ATP synthase rotates around, and that eventually combines ADP and any free-flowing phosphates into ATP. Oh my. Oh, wow. Okay, sorry. My thing was lagging for a second. All right, so now we're gonna go over photosynthesis. Like I mentioned previously, there is a lot more material that happens that you could see in these chapters. However, we're trying to speed going over the main materials. So for chapter 9, obviously there's a lot more that you need to know, but yeah. All right, so chapter 10, photosynthesis. So photosynthesis, you have the light reactions, which is the initial reaction. Solar energy gets turned into chemical energy. And then it's driven by light energy captured by chlorophyll, consumes water, produces oxygen, ATP, and NADPH. Once again, the electron carriers are here as well. And then Calvin-Benson cycle, which is the carbon fixation reactions. It doesn't use light directly, but it uses that ATP and electron carriers from the light reaction in order to eventually produce sugar. So definitely memorize this equation because you're going to have to know it. They'll test it, they'll be like, is this the equation for photosynthesis or is this the equation for ATP, etc. So know the plant wall structures. I'm not going to go over it as much, but definitely know definitely know the structures of the plant cell because for example, the chloroplast is extremely important because that's where the photosynthesis reactions take place. So know just where everything is in relation to one another. So like I mentioned, the chloroplast is the main important part for photosynthesis, as within the chloroplast is where photosynthesis actually happens, reactions happen, and eventually the light independent reactions happen. So we'll talk about the light reaction. So here it's a similar process. It's in a way it's kind of a similar process to the electron transfer chain. So first, as we mentioned before, water is consumed. As you see here, water goes through the electron transfer chain. So water is consumed, and then water gives 2 electrons to P680. So what P680 is, it's an electron acceptor within these photosystems. So it's an electron acceptor within these photosystems. And here's another thing to keep in mind of. Photosystem 2 comes first before photosystem 1. I remember in previous years, there was a question on an exam where they mentioned what's the pathway for the light reactions. Some of them say photosystem 1 first, then electron transfer chain, then photosystem 2. That's wrong. Photosystem 2 comes first. So photosystem 2, electron transfer chain, then photosystem 1. So keep in mind, photosystem 2 comes first. So water gives 2 electrons to P680. So what P680 does, it accepts the electrons and it energizes another electron. So it energizes one of the electrons it takes in, and then that electron goes through the electron transfer chain. As it goes through the electron transfer chain, the similar way that it happened in ATP, as it goes through the electron transfer chain, a proton gradient is created. ADP combines with a phosphate to produce ATP. Then that electron goes to P700 in photosystem 1, which is another electron acceptor. That electron acceptor, same process, it energizes another electron. That electron goes through ferredoxin, which then goes through NADP plus reductase, which combines NADP and a hydrogen ion to create NADPH. So here's just another way as you can see it. Hydrogen ions send 2 electrons through photosystem 2. that electrons go through further complexes. And then as it goes through each complex, it creates it creates a proton gradient, as you see here. Then electrons go to photosystem 2, ferredoxin, NADP plus reductase to create NADPH. And over, as you see here, you have an extreme proton gradient here. You need to get rid of that proton gradient. Hydrogen ions go through ATP synthase to produce ATP. All right, so does anyone want to answer? You could just type in chat, or you could type directly to me if you guys don't want to answer like publicly. All right, so 2 people responded, and yeah, both people are correct. So light energy gets absorbed, it excites an electron, goes through the electron transfer chain, creates a pH gradient. That pH gradient, as oh, my thing is like, oh, that pH gradient, as you see here, then goes through ATP synthase to create ATP. So yes, the correct answer is D. Another one. BBB, correct. So electron transfer chain phosphorylation provides the Calvin cycle with ATP and NADPH, which is the electron carrier. All right, so good job. All right, now we're going to talk about the Calvin cycle, which is the light independent reaction. Once again, definitely know your formulas. So how the Calvin Benson cycle, how was it even found? So the experiments. So you had chlorella cells. They were exposed to carbon dioxide, which were radiated. So this was 14 carbon dioxide. So the first stable compound, which is 3PG, a 3-carbon sugar, appeared after a few seconds of exposure with carbon dioxide. So through further and further exposure, by increasing the length of exposure to carbon dioxide, they're able to actually figure out what compounds were created in the Calvin cycle. So chlorella was how they figured out 3PG and all the other compounds that are part of the Calvin cycle. So as you see here in the Calvin cycle, you take in carbon dioxide from the atmosphere, and then eventually you produce sugar. So carbon dioxide gets taken in, and then after carbon dioxide gets taken in, you have 3PG. Then ATP gets used up, 12 ATP gets used up. So that 3PG, 12 ATP gets used up, and NADPH, here's where they give their electrons. 12 3PG turns into 12 G3P. Out of that 12 G3P, 2 of those are used to create more sugar, while the other 10 G3P is used to regenerate Ru-MP, which then turns into Ru-BP and continues the cycle. So you have 6 carbon dioxide and 12 3PG are formed. But from the 12 3PG, only 2 of them, all 12 turn into G3P, but only 2 of them eventually turn into sugar. And out of the 12, 10 of them is used to regenerate the Calvin cycle from G3P to Ru-BP. So you get 6 carbon dioxide, 12 3P, 2 G3P gets used for sugars, another 10 gets used to regenerate the cycle all over again. So another question, which of the following occurs during the light independent reaction? This should be pretty simple. B. Yeah, correct. Carbon dioxide is eventually converted into sugar, which is then used by the plant for obviously glycolysis. All right. As you saw, that was very quick. So now we're going to move on to the cell cycle, cell division. This is a long chapter going over mitosis and meiosis. So 3 main reasons for cell division. 1, reproduction. Obviously, animals, organisms need to reproduce. They could reproduce through various methods, but one of the main reasons for cell division is reproduction. Another reason is growth, and then another reason is certain animals are also able to regenerate parts of themselves. So reproduction, growth, and regeneration are the 3 main reasons for cell division. So the general steps for cell division. So you have a reproductive signal, which initiates the cell division. You have replication of DNA, segregate, the DNA gets distributed into 2 new cells, and lastly, cytokinesis, where the 2 new cells are separated. All right, so first we're going to start with prokaryotic cell division as it's the simplest. So prokaryotic cell division is also known as binary fission. So it's a very similar process to mitosis, not as complicated, but very similar. So DNA replication begins at the origin of replication, also known as ori, at the center of the cell. The chromosomal DNA replicates and the cell starts to grow. Then eventually the daughter DNA separate, led by the region, which includes the origin. So the cell begins to divide, and eventually cytokinesis is complete. Two new cells are formed. So it's prokaryotic cell division is very simple. DNA replicates, the cell starts to grow, the cell starts to pinch in the middle at the ori site, and then cytokinesis is complete. So we just went over this, but just to make sure you guys are paying attention. How do bacterial cells reproduce? Binary fission, correct. All right, good job, everyone. All right. This one I didn't go over, but this is just to test your general knowledge. Bacteria typically have blank, whereas eukaryotes have blank. Oh, hey, we've got some differing responses. So we have A's, B's, C's. The correct answer is A. So as you see here, let's see if I could go back yet. Here, the chromosomes for bacteria are circular. The chromosome for eukaryotes are linear. As you see, they create the common X shape that we see. So obviously, the genetic material itself obviously does appear to be circular when you look deep in, but the general shape of chromosomes are linear, whereas in bacteria, they're circular. So bacteria typically have one chromosome that's circular. Eukaryotes typically have many chromosomes that are linear. So the correct answer was A. So now we're going to go over eukaryotic cell division, also commonly known as mitosis. But first, we need to go over the eukaryotic cell cycle, where you have the interphase and then eventually mitosis. So first, you have the G0 phase. G0 phase is where if they notice any like defects with the cell is arrested at the G0 phase. So if the cell isn't supposed to divide any further, it's arrested at the G0 phase and can't go any further. Then the G1 phase is where chromosomes are single unreplicated structures. Then DNA synthesis happens where the DNA is replicated. Sister chromatids still remain together though during the S phase. Then the G2 phase is where the cell starts to prepare for mitosis. So it synthesizes structures in order to move the chromatids, such as the microtubules. And then eventually mitosis happens. So how does a nucleus in G2 differ from a nucleus in G1? Got a few answers. So the correct answer is A. G2 nucleus has double the amount of DNA because it's G1 synthesis, then G2. I saw a B, but the reason it's not B is because DNA synthesis occurs after the G1 phase. Um, as you see here, DNA synthesis S occurs after the G1 phase. So G1, S, G2, and G2 nucleus has double the DNA of G1, as that's when it starts to prepare for mitosis. And now we're going to go over mitosis. All right, so as we just mentioned, there was interphase. During prophase, the chromosomes become visible, and then the centrosomes move to the opposite poles. The centrosomes are the compounds or the materials that contain the microtubules in order to pull everything apart. So the centrosomes start to move to either end of the cell. During pro-metaphase, the nuclear envelope, as you see here, starts to break down. As the nuclear envelope starts to break down, the kinetochore microtubules appear and obviously connect the kinetochore. The kinetochore is the center part of the chromosome, starts to connect the kinetochore to the poles, and then chromosomes are gradually pushed towards the equatorial plate, which is metaphase. Metaphase, they move to the equatorial plate, or also known as the metaphase plate. As they move to the equatorial plate, then anaphase happens. During anaphase, the daughter chromosomes are split apart and move towards each side of the pole. After they move towards each side of the pole, then telophase happens, where the nuclear envelope starts to reform around each side, each chromosome. So the nuclear envelope starts to form around the newly split apart chromosomes, and then cytokinesis happens, where the cell breaks apart, and now you have two daughter cells. So which statement about mitosis is true? A. Yep. So I'm sure you guys heard of 2N and N. So mitosis, you have 2N cells, and then that 2N cells produces daughter cells that are also 2N. Meiosis, you have 2N or diploid cells, and that produces haploid or N daughter cells. So that's one of the main differences between mitosis and meiosis. So yes, the correct answer is A. So now I'm going to talk about the mitotic spindle. So you have polar microtubules. Polar microtubules extend from the spindle poles and they connect. They overlap in the center and they make sure the spindle poles kept apart because you don't want the poles to be near each other. You want them to be on each side so that the chromosomes are able to split evenly apart and you're able to produce two daughter cells with the same DNA in each cell, obviously. And then kinetochore microtubules attach to the microtubules within the centrosome in order to they attach to the kinetochores within the they attach to the kinetochores within the sister chromatids of the chromosomes, and then that's what's able to pull apart the chromosomes in order to make sure that you have in order to make sure you have the same amount of DNA on each side. You don't want one cell that has more chromosomes than the other cell. So how do these cells actually separate? So during prophase, the sister chromatids are held together by a molecule known as cohesin. In prometaphase, most of the cohesin is removed except for some at the centromere, as you see here, and that's what helps create that X shape. So at the end of metaphase, when we're going into anaphase, once it starts to split apart, a cyclin CDK complex activates an anaphase promoting complex, or APC. That APC then activates separase, another enzyme. That separase eventually removes the remaining cohesion at the centromere, which allows the daughter chromosomes to actually separate during anaphase. So now we're going to go over meiosis. So meiosis is the production of daughter cells, or 1N haploid daughter cells, gametes from 2N diploid cells. So meiosis produces sperm and egg cells. So it consists of two nuclear divisions, meiosis 1 and meiosis 2. DNA replication only occurs once, and the products are different from the parent cell. We're not creating direct copies of the parent cell. So the good thing about meiosis, not even almost, that basically is exactly the same steps as mitosis, but slight differences. One difference here, mitosis, the homologous chromosomes line up at the metaphase plate independently. So as you see here, they're not paired. So M1, M2, where we say M is the maternal, P is the paternal. As you see in mitosis, they line up independently. But during meiosis, they're paired up. Now, what's the reason for them being paired up? Crossing over. Homologous chromosomes need to pair up during meiosis so that there could be crossing over between the chromosomes so that they could create different DNA. That's the reason why your person's child is not exactly a replica of the father or the mother, or two siblings are not the same. It's because of crossing over that happens during metaphase. So the homologous chromosomes must be paired up during meiosis. So I'm not going to go over each phase again. Hopefully you guys know it, but it's the exact same. Prophase, prometaphase, nuclear envelope starts to break down, the chromosomes start to combine during prometaphase/metaphase. As you see here, the chiasma is the location where crossing over occurs. So as you see here, during late prophase, prometaphase, crossing over starts to occur. Metaphase, they line up at the plate. Anaphase, they separate. Telophase, nuclear envelope breaks down. Now you create two daughter cells. So after you create two daughter cells, the same thing happens again though. Prophase 2, metaphase 2, anaphase 2, telophase 2, and now you create parts. Now you have four daughter cells each containing half the genetic material of the original parent cell. So each cell is now haploid. So differences between meiosis 2 and mitosis. So before meiosis 2, DNA replication does not occur, because if DNA replication occurred, then you would just create two end cells. So DNA replication does not occur before meiosis 2. And once again, in meiosis 2, the sister chromatids themselves are also not identical because crossing over happens. One chromosome could have more of the mother's genes, one chromosome could have more of the father's genes. So there are other methods of sexual reproduction where fertilization and meiosis alternates. So definitely know the other life cycles that happen. So for example, certain fungus, the mature organism is haploid, but the zygote is diploid. So it's the opposite of humans, where for humans, or where it's not the opposite, it's like slightly the opposite. Where humans, obviously the mature organism is 2N, zygote is also 2N, but the gametes and are N. But here, the mature organism is still haploid. Only the zygote is diploid. In ferns, they have alteration of generations, where the organism passes through both haploid and diploid stages. And then for some birds, they have a diplontic life cycle, where the organism is diploid, while the gametes are the only haploid stage. So knowledge check. I did not go over this, but this should be simple. What's the drawback of asexual reproduction as compared to sexual reproduction? D, right. Asexual reproduction, you're producing an identical copy of yourself. If for some reason that identical copy is not able to survive because of a change environment, etc., those cells are going to, the offspring are going to die off. There's less variation. They're exact copies. All right, meiotic errors. So sometimes there are errors that occur where, as we were talking about, that's the importance of the spindles to make sure that during anaphase, the chromosomes are able to split apart properly. However, sometimes there are errors that occur. So one of them, as people know, Down syndrome, also known as trisomy 21. So in humans, we have 23 homologous chromosomes. One of them is obviously your XX gene or XY gene. So non-disjunction can occur where during anaphase, both homologous chromosomes go to the same pole. As you see here in meiosis 1, both homologous chromosomes go to the same pole. Then in meiosis 2, you have an extra chromosome, while in one of them you have no chromosomes. Then when the cells are fertilized by sperm, you have monosomy, where you have a cell with only one chromosome, and trisomy, where you have a cell with three chromosomes. So trisomy 21 is what causes Down syndrome. And so the reason why trisomy and monosomies are actually really common. But the reason why we only, like, this doesn't affect the offspring as much is because uh most uh fertilized cells that suffer from monosomy or trisomy, they are lethal, and that's what causes miscarriages majority of the time. So trisomy 20 main one, where if there is a trisomy, zygote is able to actually survive and eventually become a baby. But most monosomies or trisomies are lethal, and that causes miscarriages. So trisomy 21 is an exception, where chromosome 21, having an extra chromosome, obviously it does create a genetic deficiency, but they're still able to survive. But most of them are lethal, unfortunately. So I just went over this, so I'll skip this, but many chromosomal abnormalities aren't observed in the human population because they are lethal and cause spontaneous abortion of the embryo early in development. So now we're going to talk about inheritance. So as you guys know, you have heterozygous genes and homozygous genes. Or heterozygous alleles and homozygous alleles. So heterozygous alleles are alleles that carry this same gene heterozygous alleles, same gene at same locus, carry different alleles. So you have here in this case uppercase R, lowercase r. Homozygous, you carry the same allele. So you can have two uppercase R's, two lowercase r's. As we know, for uppercase R, we consider that dominant. Anything that's uppercase, we consider dominant. Anything that's lowercase, we consider recessive. So heterozygous, one of each. Homozygous, you have either two dominant alleles or two recessive alleles. So as we mentioned before, you have dominant genes and recessive genes. Dominant genes are the genes that are expressed. Recessive genes are the genes that you need two alleles in order to show it. For dominant, if you're heterozygous, the dominant gene is still going to be shown because you have at least one of the dominant allele. So in order for the recessive gene to be shown, such as here, for example, no cleft or no dimples or gray blue eyes, in order for that to be shown, you need two of the recessive alleles. For the dominant gene, you just need one of the dominant alleles. So only one of the two has to be dominant. Recessive, both have to be recessive. So now we can get into genotype versus phenotype. Genotype is your gene. Phenotype is what's actually shown on the surface. So genotype codes for the phenotype. What you see on the face, that's kind of how I think about it. So for example, your genotype could be uppercase A, and your phenotype would be brown eyes. Your genotype could be lowercase a, your phenotype would be blue eyes, for example. So phenotype is the physical way of seeing it. Genotype's the actual allele combination. Punnett squares. So for Punnett squares, you have two parents. Punnett squares, you could have multiple Punnett squares, but this is a monohybrid cross. So in a monohybrid cross, here a homozygous dominant parent, uppercase A, homozygous recessive parent, uppercase A, or lowercase a. In order to do a Punnett square, you put one allele on one square, another allele on another square, and then on the opposite side, you put the other two alleles of the parents. And then it's just you combine both alleles. So here, if you have a homozygous dominant parent and a homozygous recessive parent, all your children are going to be heterozygous. Case A, lowercase a. However, what happens if you self-pollinate, which as we saw in like Mendel's crosses, for example, if you have two heterozygous parents that self-pollinate or combine, the second generation creates what's known as the genotypic and phenotypic ratio, which is pretty common for monohybrid crosses, 1:2:1 or 3:1. So this is the case if you have two heterozygous parents for a monohybrid cross. 1:2:1 means you have one homozygous dominant, two heterozygous, one homozygous recessive, while your phenotypic ratio is 3:1. Because as I mentioned before, as long as you have one allele that's dominant, if you're heterozygous, as long as you have one allele that's dominant, you're going to show the dominant trait. So your phenotypic ratio is 3:1, but your genotypic ratio is 1:2:1. So sometimes these ratios will help because some of the questions we'll talk about for dihybrid crosses, especially they'll help, which I'll go over a bit later. Some of the questions are gonna go over you have two heterozygous parents for two traits. What's the chance for one of the kids to be homozygous dominant? So if you know the ratios ahead of time, you don't actually have to create the Punnett square during class. And for dihybrid crosses especially, it'll take a lot more time. So first I'll go over Mendel's monohybrid crosses. So Mendel wanted to test if when two strains of peas, if contrasting traits are bred, if their characteristics will be completely blended in succeeding generations. So they had a true breeding round seed, so which is in this case homozygous dominant, a true breeding wrinkled seed, in this case homozygous recessive. But the hypothesis was rejected because there wasn't a blending of characteristics, and eventually F2 seeds from the F1 plant, the recessive trait can reappear. So here, the F1 generation, you see they all show dominant traits in that they're all round. However, once they self-pollinated, you were able to see a F2 seed. So there's no blending of traits. You could have a recessive trait not appear in one generation, but appear in the next generation. So this is known as the law of independent assortment, where you're getting one allele from one parent, another allele from another parent. And then law of independent assortment is that each allele separates independently of one another. Just because you have two homozygous dominant alleles does not mean your children will also have two homozygous dominant alleles. That's the point of the monohybrid cross. Each allele separates independently of one another. Law of segregation is that you gain one allele from each parent. So in this case, you gain one allele from your mom's side, one allele from your dad's side. So dihybrid crosses, two traits. So when you have a dihybrid cross for two traits, it's the same process here where they took the two traits were the color, yellow or green, and the structure of the seed, spherical or wrinkled. So it's the same process. You had homozygous dominant for both traits, so spherical yellow, homozygous recessive for both traits, wrinkled green. So when they combined both parents, you obviously had children that were all spherical yellow, but they're heterozygous for both traits. Now, when you combine two heterozygous individuals, you get what's known as the 9:3 to 3:1 ratio. So dihybrid cross, two heterozygous parents, heterozygous for both traits, you get the 9:3 to 3:1 ratio. 9 homozygous dominant for both traits. 3 dominant for one trait, recessive for the other trait. 3, once again, dominant for one trait, recessive for the other trait, and then one of them is recessive for both traits. So the way that I do these dihybrid crosses, I kind of create a web basically. So can you guys see my mouse? Yeah, right. So if you guys can see my mouse, you take the first allele of the first trait and then create a web with this first allele of the second trait. Then this allele, once again, law of independent assortment. This allele could also go with this allele. So create another web with this guy, create another web with the second allele of the first trait, first allele of the second trait, second allele of the first trait, second allele of the second trait. So basically you're creating 4 lines. 1 to 3, 1 to 4, 2 to 3, 2 to 4. And then same thing here, 1 to 3, 1 to 4, 2 to 3, 2 to 4. As long as you're able to create that web, then you're going to be able to, you have your 4 combinations. You're going to be able to put your 4 combinations on the top, your other 4 combinations on the left, and it's the same process, you just combine the letters that you see. So for dihybrid crosses, create the web between each allele. So 1, 3, 1, 4, 2, 3, 2, 4. So different alleles or variations of alleles arise due to mutations. Random mutations can cause more than 2 alleles to be present in a population. However, one individual can only carry 2 alleles. Multiple alleles increase the number of potential phenotypes. A wild type allele, so wild type refers to the allele that's present the most in a population. Mutant alleles are present the least in a population. So now we're going to talk about gene interactions with one another. So you can have complete dominance, incomplete dominance, and co-dominance. So complete dominance is what we mentioned before. If you have a dominant allele, as long as you have one of that dominant allele, that dominant allele is the one that's going to be shown. Incomplete dominance is where neither allele is dominant to one another, so you get a mixture of both alleles. As you see here, you have a light blue child generation. Co-dominance, both alleles are somehow dominant with one another, so now both alleles are represented in the child generation or the F1 generation. So there's also something known as polymorphism, where some genes can have more than 2 alleles, but that depends on the environment and depends on temperature. For example, here you have a dark gray allele. So all of these genotypes have a C, where C is the color in dark gray. You have here a chinchilla, where CHD in this case refers to chinchilla, that's the color. So here CHD appears to be dominant to C. Then you have point restricted. Point restricted means that this specific rabbit, they're temperature dependent. So they have a CH, H stands for Himalaya in this case. So the fur coat that they actually show is dependent on the temperature of the area that they're in. So it's point restricted. And then lastly, you have here an albino where you have 2 small C's, and that's completely recessive. Once again, as we mentioned before, you can have incomplete dominance if both traits are somehow if both alleles aren't completely dominant with one another. You can have a purple eggplant, a white eggplant combined, you create a violet. And then when they cross with themselves, you can have a 1 to 2 to 1 ratio. As you mentioned before, 2 heterozygous monohybrid creates a 1 to 2 to 1 ratio. 9:3 to 3:1 ratio for dihybrid. So now we're going to talk about DNA. So Frederick Griffith's experiments, genetic transformations. So the bacterial cells can genetically transform living bacterial cells. So know some experiments because some of the experiment results will be tested on the actual exam. So Frederick Griffith determined that a transforming principle from dead cells of one strain can actually produce a heritable change in the other strain. So as you see here, you have a living S strain, which is virulent. You inject the mice with it, the mouse dies. Living R strain, non-virulent, inject the mouse with it, nothing happens to it. No bacterial cells found in the blood. You can kill the S strain bacteria by heating. So completely kill the S strain bacteria and inject it into a mouse. The mouse is still healthy. But once you mix the dead S strain cells with non-virulent R strain cells, so as you see here, the R strain cells do nothing to the mouse, and the S strain bacteria is dead. But once you mix the dead S cells with the non-virulent R strain cells, the mouse is still able to die, and they're able to find living S strain cells in the blood. So it shows that the cell is capable of genetically transforming another cell. So dead bacterial cells can transform living bacterial cells. So now we're going to talk about the Hershey-Chase experiment. This is how they figured out whether DNA or protein was the genetic material. Because a lot of people actually assume protein to be the genetic material because proteins are more complex. So the way they figured it out was first they put DNA. So the DNA was radio labeled with 32P, while the bacteriophage proteins were radio labeled with 35S. So the DNA was radio labeled with 32P, and then bacteria were both put in, and then they were centrifuged, and then a pellet was created. So after that pellet, they found that the phage's DNA was entered the bacteria. So the phage's DNA was not found in the bacteriophage anymore, or here, but it was actually found within the actual bacteria. On the other hand, when they did it with protein, the bacteriophage protein, which was labeled with 35S, when they combined everything together, they found that the protein that was radio labeled did not enter the bacteria. It was still within this phage. So the DNA was able to enter the bacteria, and they were able to notice that because it was radio labeled, while here the bacteria did not have any radio labeled protein in it. So through that, the Hershey-Chase experiment showed that the genetic material that's able to infect bacteria, or our genetic material that's able to create new life, etc., is DNA. So the structure of DNA, so you have purines and pyrimidines. Purines are adenosine and guanine. Pyrimidines are thymine and cytosine. So A plus G is always equal to T plus C. And your two combinations are A and T, G and C. For RNA, it's A and U, G and C. So the amount of adenosine you have will always equal the amount of thymine you have. The amount of guanine you have will always equal the amount of cytosine you have. And adenosine and guanine are your purines, thymine and cytosine are your pyrimidines. So if a sequence in one strand of DNA is 5 prime, this DNA strand 3 prime, what is the sequence in the complementary strand? Correct, is D. Because DNA strands are anti-parallel to one another, so the complementary strand is going to go from 3 prime to 5 prime, and the way you figure out the complementary strand, it is the opposite. Adenosine is combined with thymine, so replace all the A's with T's. Guanine is combined with cytosine, so replace all the G's with C's, and then vice versa. So, yep, the correct answer is D. All right, another question. 30% of the bases in a sample of DNA is adenine. What percentage of cytosine is present in the DNA? Oh, I see a lot of C's, but C is actually not the correct answer. B is the correct answer. So this is another way they try to trick you, because remember what I said? Uh, adenine and thymine are the combinations. So if you have 30% adenine, you have 30% thymine, not 30% cytosine. So you have 30% adenine, you have 30% thymine, 30 plus 30 is 60, you have 40% DNA left, 40 divided by 2 is 20. So now you have 20% cytosine, 20% guanine. So the correct answer is B. So a lot of times that's how they're going to try to trick you. They'll have, yeah, a lot of times they'll try to trick you. They'll have combinations that aren't together, in which case you'll think, oh, since it's 30% adenine, it'll be 30% cytosine. But you have to remember, adenine and thymine are the combinations, are the two that are connected, and cytosine and guanine are the two that are connected. All right, so structure of DNA. Rosalind Franklin was able to figure out the structure of DNA through X-ray crystallography. So through X-ray crystallography, DNA was able to be found out that it has a double helix with a sugar phosphate backbone on each strand. And then James Watson and Francis Crick, they used model building and combined prior knowledge to determine the actual structure of the DNA, where DNA is double-stranded, it is right-handed for the most part, it's anti-parallel. One strand is 5' to 3', other strand is 3' to 5'. And then you have nitrogenous bases that are exposed in the major and minor grooves. So just went over this. Which of the following models describes the structure of DNA? C. Yeah, double-stranded antiparallel. Good job. All right, so the function of genetic material, what's the importance of DNA? DNA stores genetic information. It's susceptible to mutation. Obviously, some mutations are bad, but at the same time, some mutations are good. It's precisely replicated in cell division, and your DNA is expressed as the phenotype. So now we're going to go over DNA replication. So primase binds to the template strand. So if your template strand is 5' to 3', the RNA primer is going to be the opposite, so here the template strand, the primer is reading the DNA towards the left. So the primase is reading the DNA from 3' to 5'. So the RNA primer is the opposite. The RNA primer is being produced 5' to 3'. So primase binds to the template strand, synthesizes an RNA primer, and then when the primer is complete, the primase is released. So after the primase is released, DNA polymerase then binds to the template and it starts to create a new strand of DNA. And then DNA polymerase is able to, so the primer, the only point of the primer is as a starting strand. As you see here, this green part, it's only the starting strand. The primer doesn't actually continue on the whole way. It's just as a way of letting the DNA polymerase know where to start from. So the DNA polymerase starts to elongate both strands. So you have the leading strand and the lagging strand. And while DNA helicase unwinds the double helix, and you have DNA polymerase on both strands that are replicating the DNA. So the leading strand is able to do it continuously because here it's going from the 5' to the 3' direction. So it's able to do it completely continuously without any issue. However, for the lagging strand, it's not able to do it continuously. So because it's not able to do it continuously, we have what's known as Okazaki fragments. So the primase synthesizes a primer, then the DNA polymerase goes there and creates the Okazaki fragment, and the lagging strand keeps on continuing the process of primer Okazaki fragment, primer Okazaki fragment. And that's how for the lagging strand it's able to actually replicate the DNA, whereas for the leading strand it's able to do that seamlessly. So here, synthesis of the leading strand is continuous, while the lagging strand is synthesized as Okazaki fragments. So the replication fork is the point where the DNA helix starts to unravel. As the replication fork starts to grow, as you see here for the lagging strand, you have primase puts the RNA primer, then that primase leaves, DNA polymerase fills in the gap between the primers. So the process will continue here. There'll be a primer somewhere here. The primer will leave, DNA polymerase will come in and fill in that gap with the Okazaki fragment. So here, it fills in that gap, And so in order to get rid of the RNA primer, um, DNA polymerase 1 replaces the primer with DNA, and then after it replaces the primer with DNA, you have a gap in between. So because you have a gap in between, the DNA polymerase leaves, ligase comes in. DNA ligase comes in and then closes that gap. So you have once the RNA primer creates a gap between the Okazaki fragments, RNA primer is converted into DNA by DNA polymerase 1, but now there's still a gap. So in order to get rid of that gap, DNA ligase comes in. Ligase is the glue. Ligase joins in those fragments. So DNA polymerase 3 can make mistakes in replication, and as a result, DNA can be damaged in living cells. So the cells have repair mechanisms, which are proofreading, mismatch repair, and excision repair. So for proofreading, if bases are paired incorrectly, the nucleotide is removed, and then DNA polymerase then comes in and just adds a new one. For mismatch repair, a mechanism is able to detect mismatched bases. And so this is before the strand has been modified. So the bases are mismatched, they're wrong. The bases are eventually removed, and then DNA polymerase can put in the correct bases. And then excision repair. For excision repair, enzymes scan the DNA for any damage. So for excision repair, enzymes are able to detect that this DNA, the yellow, it has a damaged base. So because of that, enzymes themselves excise the DNA, replace it, and then DNA polymerase 1 adds the correct bases. All right, and now we're gonna go from gene to protein. So the central dogma of gene expression, DNA turns into RNA, which turns into protein. DNA to RNA is what's known as transcription. RNA to protein is what's known as translation. And then expression of the gene obviously yields a protein. So there are some exceptions where some viruses, also known as retroviruses, they can use RNA to encode information. So RNA gets transcribed into RNA and then translated into polypeptides and vice versa. But some viruses can also convert RNA to DNA. So DNA gets transcribed into RNA, but that RNA can be reverse transcribed into DNA. And so the transcription and processing process all occurs within the nucleus. So transcription, where the DNA gets transcribed into pre-mRNA, and then that pre-mRNA gets processed into mRNA, and then eventually gets translated outside the nucleus into creating the polypeptides. So RNA differs from DNA, in that RNA has one polynucleotide strand. It uses ribose instead of it uses ribose instead of deoxyribose. So what that means is that it has an oxygen. As you see, deoxyribonucleic acid, DNA does not have an oxygen on the ribose. RNA has an oxygen on the ribose. Another thing is the base, as I mentioned before it uses uracil instead of thymine. So for DNA, ATCG is the combination, while for RNA, AUCG is the combination. And you have three types of RNA in protein synthesis. You have mRNA, tRNA, and rRNA. mRNA is messenger RNA, tRNA is translational RNA or transcriptional RNA, rRNA is ribosomal RNA. Um, oh, all right. So messenger RNA. So the point of messenger RNA, it carries the transcript of the DNA sequence to the site of protein synthesis at the ribosome. It's similar to the coding strand of the DNA, but it's complementary to the template strand of the DNA. So RNA is going to be the same exact thing as your coding strand, except T is replaced with U, but obviously it's complementary to the DNA strand. And it does undergo post-transcriptional modification before translation. So here's another way they'll try to trick you. They'll give you the DNA of the coding strand, right? So the reason it's called the coding strand is because your RNA is essentially the exact same code as the coding strand. So one way they'll try to trick you on the test, they'll give you the DNA of the coding strand and they'll tell you what's the mRNA. Remember, the mRNA is the exact same thing as the coding strand, except T is replaced with U. So it's the same direction, same exact, almost the same exact base, except the uracil is replaced with thymine. The mRNA is complementary to the template strand. Yeah. So if your coding strand is 3' to 5', your mRNA is 3' to 5' as well. But if the template strand is 3' to 5', then your mRNA is 5' to 3'. So the template strand is what your messenger RNA is the opposite of. So like I said, they'll try to trick you. They'll give you the coding strand, and then they'll give you the coding strand and to tell you what's the mRNA. People will put the opposite of the coding strand when that's not the case. mRNA is equivalent to the coding strand, except it has uracil. mRNA is the opposite of the template strand. So if they give you the template strand, put whatever the opposite is. If they give you the coding strand, same exact thing, but with uracil. Oh, and I saw someone put a question about is there a way we could share the recording? I think the recordings will be on the Learning Commons website. All right, now transfer RNA. Transfer RNA is what actually translates the nucleic acid sequence on mRNA to create the polypeptides. So you have 20 transfer RNAs, one for each amino acid, and each transfer RNA has an anticodon that's complementary to the codon sequence on the mRNA. And the codon is 3 pairs. So the tRNA interacts with mRNA and rRNA during protein synthesis. So tRNAs help create the polypeptide. Lastly, you have ribosomal RNA. Ribosomal RNA are transcribed constantly in the nucleoli. They form the bulk of the ribosomes and the cytoplasm to where protein synthesis occurs. It provides structural support and also acts as an enzyme, ribozyme, which catalyzes the peptide bond formation in translation. So peptide bonds are obviously how proteins are able to connect with one another. All right, so transcription materials. So you need the DNA template, obviously, for base pairing, nucleotides, ATP, GTP, CTP, UTP, ATP, adenosine triphosphate, guanine triphosphate, cytosine, and uracil. And then you need your RNA polymerase enzyme. So transcription has 3 processes: initiation, elongation, and termination. So initiation is where you open the DNA and bring in RNA polymerase. Elongation is when you add in nucleotide length. So you lengthen the nucleotides, and then or you lengthen the mRNA by adding nucleotides, and then termination is when it actually releases the mRNA. So you have the promoter region. So what's the point of the promoter region? So the promoter region is a sequence of nucleotides that tell the RNA polymerase where the gene begins, what strand is the template, and what direction the RNA polymerase should move. So like you mentioned here, you have the template strand, in this case is 3' to 5'. Your RNA transcript is the opposite, 5' to 3'. This RNA transcript is equivalent to this strand. So RNA polymerase binds to the promoter region of a gene. So as you see here, RNA polymerase binds to the promoter region of a gene. And as it binds, the DNA also starts to unwind. And then here is where the initiation site is. So the initiation site tells the RNA polymerase, okay, from here on out, now I'm going to start creating the mRNA. And then now you also have a termination site. The termination site is a set of nucleotides that tell you, hey, stop creating your mRNA here. So now elongation occurs. So this is the site of the promoter. Transcription occurs in this way. RNA transcript is exiting that way. So here the template strand is being read in the 3' to 5' direction. So which is this guy right here, 3' to 5', while your exiting RNA transcript is synthesized in the 5' to 3' direction. The RNA transcript is anti-parallel to the template strand. So as the RNA polymerase starts to go through, it creates an RNA transcript from the template strand. and eventually it reaches the termination site. Sorry, I forgot the name. Eventually reaches the termination site. So the termination site is a set of certain bases which the RNA polymerase is able to recognize, and then because it's able to recognize it, it knows, okay, now I'm not going to create any more mRNA. I'm not going to add any more nucleotides to the mRNA. So sometimes the RNA polymerase just falls off once it reaches the termination site, or another protein comes and pushes the RNA polymerase off the termination site. So post-transcriptional processing and modification. So obviously it is processed before leaving the nucleus because they wanna the cell wants to make sure that you're not sending improper mRNA out, which will then create improper proteins. So you have exons and introns. So introns are your non-coding regions or sequences, while your exons are your what's actually expressed. While exons are what's actually expressed. So basically what happens, so introns and exons appear in the pre-mRNA, but in the mRNA, only the exons appear. The introns are excised out. So the sequence of the introns are able to be recognized by the enzyme. So because it's able to be recognized by the enzyme, it cuts it off. Once it cuts it off, only the coding regions of the pre-mRNA are actually kept on to the mature mRNA. And then onto the mature mRNA, you have a 5' GTP cap and then a polyadenosine tail. So the 5' GTP cap allows for the mRNA binding to the eventual ribosome, and it also protects the mRNA from being digested by ribonucleases, while the 3' poly A tail stabilizes the mRNA. And that's also another way the ribosome is also able to identify, hey, I'm not going to keep creating a protein just through through 3 bases of AAA. So the poly A tail stabilizes the mRNA, while the 5' cap protects it from being digested and allows it to bind to the ribosome. And now comes translation. So mature RNA travels from the nucleus to the cytoplasm. A cap binding protein complex binds to the 5' end of the mRNA in the nucleus. The 5' end is the GTP cap, which we added as well. So these cap binding proteins lead the mRNA through the pores of the nucleus, while unprocessed pre-mRNA still remains within the nucleus. So 2 key events happen during translation to ensure that the protein is actually synthesized. You have codons that need to be read correctly by the tRNA anticodon, and the tRNA obviously has to bring over the correct amino acid. So here. So you have 2 subunits, the large subunit and the small subunit. And within the large subunit, you have an E site, P site, and A site. So when not in translation, the subunits exist separately, but during translation, the subunits combine. So AUG is your start codon, and so E is your exit site. So the polypeptide is being created this way, where you see my mouse is being created that way. So the codons will slowly move towards the right, while your polypeptide will be exited towards the left. So E is your exit site. P is where amino acids are added to the growing polypeptide, and E is your amino site, which presents the next set of 3 bases. So your A site presents the next set of 3 bases, and it binds to the anticodon of the charged tRNA. So translation also has 3 steps: initiation, elongation, and termination. So initiation is your AUG start. So once the 5' cap is bound to the small subunit keeps moving along the mRNA until it finds the start codon. Once it finds the start codon, large subunit is brought in and the tRNA is brought in. Once again, tRNA has an anticodon. The tRNA has anticodons that are the opposite of the codon. So here your codon is AUG, anticodon is the opposite, UAC. And in this tRNA, it brings in the amino acid associated with that anticodon. So your anticodon, UAC, it brings in MET. So the large subunit catalyzes two reactions. It then breaks the bond between the tRNA in the P site and its amino acid. Oh, sorry. The large subunit catalyzes two reactions. It breaks the bond between the tRNA in the P site and the amino acid, and it catalyzes the formation of a peptide bond between this amino acid and the amino acid of the tRNA in the A site. So here, P site, that's when the first amino acid is brought in. A site notices the next three codons and it starts to bring in the tRNA for the next codon. As that tRNA is brought in, over here, the large subunit combines the amino acid from the P site, creates a peptide bond between the amino acid from the P site, and amino acid from the A site. Then the amino acid from the A site moves on to the P site, while the tRNA exits from the E site. And then it's a continuous process. It continuously happens until eventually the stop codon is released. So once the A site detects the stop codon, so when a stop codon enters the A site, a release factor then binds to the A site. The release factor binds to the A site, and so the polypeptide chain is then released from the P site. So stop codon is identified, release factor binds to the A site. As it binds, the polypeptide chain is then released from the P site. And then the remaining components separate because translation is over at this point. So there's three types of post-translational modifications. Proteolysis, which the polypeptide chain is cut up by proteases. So you could have one gene that produces multiple peptides. That one polypeptide chain doesn't necessarily need to be one whole protein. It could be split up into multiple proteins. Glycosylation, where sugar is added to form glycoproteins, and then phosphorylations. So the phosphate groups are added by protein kinases, and that changes the conformation of the protein. So we're near the end, thankfully. So obviously I'll stay after 1:30. I know it's free hours, so I'll stay after 1:30 if you guys have questions. So wild type, as I mentioned before, wild type is your normal. That's just what's typically seen. You have a silent mutation. A silent mutation is where, as you see here, so once again, amino acids are three codons. A codon is made up of three bases. So an amino acid is made up by three things. So you can have a silent mutation. In this case, the mRNA is replaced. In this case, the mRNA has a strand where here you have GGA, before it was GGC over here. Before it was GGC, now it's GGA. It's translated to CCU, so it's a completely different base. However, once it's translated, you have the same exact amino acid because one amino acid can be created by multiple different codons. So that's why it's called a silent mutation. The base is changed, but you still have the same amino acid. You have a missense mutation where the base is changed, but you have a completely different amino acid, as you see here. The base is changed, now you have a completely different amino acid. You can have a nonsense mutation. A nonsense mutation is when you have a mutation that creates a stop codon early. So in this case, a stop codon was created early due to the mutation. And then you have a frame shift mutations where um a base was inserted now. Rather than something being swapped out, here a base was inserted in the DNA. So that shifts over all your codons. So now you have a completely different amino acid chain due to the shift of the um additional base that was added. And then these are some some mutations that happen. These are normal alleles. You can have a mutation, once again, as I mentioned, silent mutation. You still have a functional protein, A mutation create a loss of function where now your protein does the mutation, whether it was a deletion, frame shift, etc. Now your protein is non-functional. Or you could also have a gain of function mutation. As we mentioned before, mutations are both positive and negative. This protein now has a new function thanks to the mutation. And yeah, that's everything. Hopefully I was able to help out. No chapters. Oh yeah, so for chapter 16, I wasn't able to go over it, obviously due to the time constraints. I knew that if I had to go over chapter 16, it would be I'd go past 130. So chapter 16, just because I didn't go over chapter 16 does not mean it's not on the exam. So definitely study chapter 16, study part of the chapters that obviously I didn't go over. Um you're welcome. Thank you guys so much for showing up. This shows that obviously these study sessions are a great way for us to help you guys out. And yeah, it shows that there is a need for these study sessions. So, thank you guys for coming and showing up. If you guys have any questions for for the future exams, lab, etc., any tips for the class, definitely ask me. Um, I don't unfortunately have slides for chapter 16. I could try and find some, but unfortunately I don't currently. The slides should be available within like the next few hours. Like it'll be uploaded right after the meeting. Right after the meeting's over, the Zoom will be uploaded, the slides will be uploaded. I think Arena could tell you where to see the slides, but I'm pretty sure it's in the if you search up Queens College Learning Commons website, it should be there. The YouTube link, if you guys search up Queens College Learning Commons on YouTube, I'll type it in chat. Okay, so the slides is gonna be there. Um Queens College Learning Commons. That should be, if you search that up on YouTube, you'll find it for exam 2. And I also did an exam 3 and final review session from last semester. So you guys could also find those videos for like future study sessions if you guys want to eventually when you study exam 3 and the final. Um, Studying for such a content heavy course. So Dr. Fat does not provide a curve. If there is a curve, it would only be a maximum of like 1 or 2 points. So Dr. Fat does not provide a curve, or Dr. Garofalo. So they don't provide curves for the class. However, when it comes to actually studying for the class, I would recommend studying from the slides. And if you can try recording lecture, I know both professors are completely fine with you recording lecture. The reason I mentioned that, the textbook provides too much information where it's a lot more information that you actually need to memorize and do well in the course. Whereas, and also the slides, Dr. Fat and Dr. Ruffalo always say this, whatever is on the exam is going to be from the slides. So obviously it is a lot of information that you're gonna have to memorize. So that's why I like recording lecture. Um, when I took the class, I recorded lectures, I re-listened to the lectures as I went over the slides. Because another thing you guys might notice, sometimes the slides will only have images, right? The slides will only have images. For example, me as well. Some of my slides only had images, but it didn't have any like a text explaining it. The explanation was actually me talking about it, which is why you'll notice for Dr. Fat and Dr. Garofalo slides, some slides are going to be images, but you need someone to actually explain it to you, which is why recording lecture is something that I really enjoyed and I really needed. So obviously I listened to it at a higher speed, but having those recorded lectures, whenever there was a slide that was just an image or like certain processes example, having someone explain that to me, like re-explain that to me, really helped. So study from the slides, number one for sure. Don't study from the textbook. A lot of extra information. Slides, without a doubt, number one. Try recording lectures. Obviously, without a doubt, it is a lot of content, so do your best. Do your homework on time. Um, I think homework is worth the same as an exam or almost the same as an exam. So I don't have any past practice exams, unfortunately, but I know practice exams are typically found on like typically if you guys have a WhatsApp group chat, I know people have practice exams. The slides will be posted like once the meeting's over. So within half an hour, don't worry, the slides will be posted. It hasn't been posted yet. Um well, oh yeah, do your homework, please, because like I said, it's almost the same as an exam. So if you don't do as well, if you haven't done as well in the first exam, your homework grade could help pick that up because it's like I said, it's almost worth the same exam. Another tip, lab, definitely focus on lab. Lab is 40% of your grade. There's a lot of kids where, for example, they're maybe not the best when it comes to exam taking. However, labs through lab reports, they can actually boost their grade up significantly because lab plus homework, I believe, is around 50% of your grade. So obviously try your best on lab as well. Try your best on your lab reports. If you know that you're not maybe the greatest test taker, then trying your best on lab reports, because that's 40% of your grade. That could significantly boost you as well. There are lots of kids who did not do the best on the exams, but thanks to lab grade, they were able to get B's, A's, etc. Or if you're just wanting to pass the class, they were able to pass the class as well. So lab can significantly boost your average as well. So, don't neglect lab for the lecture, because biology is one of the only courses where lab is worth such a significant portion of your grade. So, and pay attention to both is what I'm trying to say. Both are really important. Try your best on both. Good luck on the exam tomorrow. I know it's tomorrow, so yeah, good luck on the exam tomorrow. Um, if anyone has any more questions, I'll still be here. Yeah, thank you for showing up again. Oh, also, if you guys, so the Queens College Learning Commons, I don't know how many of you guys know about it, but Queens College has what's known as the Learning Commons, which is a tutoring center, right? So the Learning Commons provides tutoring for all essentially all subjects, from CS to math to biology. So I am a tutor for the Learning Commons. So you guys, if you guys search up Queens College Learning Commons, you guys would be able to like book me for like individual tutoring appointments as well. So definitely, take advantage of that. Don't just like, you know, um, leave it all to the last minute. A lot of people, and I also help with lab as well. So if you guys have questions on lab report, just the class as well, I could definitely help with that. So definitely show up to like the Learning Commons, because we have our own times to tutor. If the times obviously fit with your schedule, definitely show up for those tutoring appointments, because that's what we're here for. So for chapter 16, I could try and add slides to, so the appointments are not on Navigate. Give me 1 second. I could search you see. Here, I'll put the thing in chat to everyone. Oh, paste. Yeah. So that's the appointment. So there's other bio tutors as well, like not just me. There's more bio tutors. So you could definitely go to them for help if you guys need help for math, psych courses, etc. Like go on their website, look. There's lots of subjects that there's lots of tutors that are there. You have to log in to that website through your CUNY. You have to log in through that website through your CUNY login. The online portal doesn't allow you to make an appointment. Oh, are you sure? Most people are able to make appointments like by logging in through like CUNY. They're able to like make an appointment with people, like you search up. Thank you. Yeah, so if it's not too much trouble, yeah, I'll try my best to look for chapter 16 slides, because obviously I'd have to create them. So I'll try my best to post that. But if not, apologies in advance, because obviously, like I said, my tutor is only until 1:30 due to free hour. So it was either for me, the option was either, make every other chapter smaller and make in order to fit chapter 16 in, or try to give as much time as I could to those chapters, because chapter 9 through chapter 14, as you guys saw, is a lot of concepts, a lot of materials, a lot of stuff to memorize. So I wanted to place emphasis on that. Chapter 16 is, I believe, gene expression, expression. So obviously I'll try my best to post some slides for that. But yeah. So yeah, if you guys have any more questions, ask away, or have a good rest of your day. Hopefully you guys do well tomorrow's exam. Carrie, if you're able to try, yeah, if you're in person, you can show up to Kylie 131 or Kylie 144. So if you show up in person, you could go to 131 and 144, and there are people that are known as front desk hosts, amazing people. They can help you try to make those appointments, and they can find out like what's wrong for you. Anyone you can email. Arena, I'm not sure if there's someone like she could email because she can't make an appointment, but if you can, try your best to like show up in person. 131 or 144, they could help you create your account, help you set up an appointment and all that. Uh, so give me 1 second. So let me scroll up. Oh, 1 second. Control C. 1, 2. Once again, uh, once the meeting's over and within like give it like half an hour to an hour, the slides will be posted to that website right there. So you guys can look at the slides on your own free time as well. You're very welcome. Please fill out the attendance form or the feedback form. It really helps me know what to improve on or, how we can make the next study sessions better. And also that shows us that there is a need for these study sessions. Um, yeah. Practice exams were easier than the real ones, in my opinion, as well. But I think for practice exams, the main point of the practice exams is just to know the structure of the exam. Oh, that's another thing. One thing you guys might notice, the exams themselves, like the questions themselves aren't necessarily like super complicated or convoluted. The questions themselves are actually really simple. It's just what is A, what is B, what is C, etc. Right? So the questions themselves are simple. It's just the amount of material that you're gonna have to memorize is the main issue. So the practice exams are definitely easier, but they just teach you the way that Dr. Fant and Dr. Garofalo go over the exams. Their exams are the questions I've always find are like super straightforward. The hard part is a lot of chapters, a lot of material they're gonna have to memorize. So reading from the slides, recording lectures, like I said, those are like the best methods for, preparing you to do well for the exams. Hmm. Of course. Also, another thing, so I know like one thing you could do for some people that like if they're running short on time and they want information in like a more concise format, a lot of people they like input like the PowerPoint slides into like Chat GPT, etc., and they create like study guides off that. So you could obviously also do that. Input your slides into like Chat GPT and then use that to help create a study guide of like what you feel are the most important topics. And then through that you get like a whole list of like what they consider the most important topics, etc. Yeah. So, all right, give me 1 second, 1 second. All right, got it. Yeah, so like I mentioned, the ChatGPT thing is really helpful because creating a study guide for everything, it's really concise and you could print it out and then, let's say you have to do some like last minute memorization, quickly scan through the study guide, look at whatever thing you find you struggle with. And then maybe you could create the study guide and then use it while listening to lecture, etc. There's a lot of different methods you could use, but like I said, slides, number one material to help you do your best. Of course. Thank you guys for being here once again. I know I've said that a lot, but thank you guys for being here and hopefully you guys do well. Hey, do you have any questions or? It's okay if you don't.